Okay, now that we know what DNA looks like and we know all the bits and pieces that it's made of and how those bits and pieces come together, now we're finally ready to talk about how does DNA make more copies of itself? Well, this process is going to be called replication because you're replicating the DNA. You're making an exact copy of it. So DNA replication is like taking your DNA, putting it on a photocopier, and pressing copy. Okay? Same idea, of course, a different process. So we're going to learn the step by step. So remember that the cell theory has 3 parts. And one of those parts was that all existing cells come from pre-existing cells. So when cells divide to make new cells, you have to make another copy of the DNA because both cells have to have a copy of the DNA so they know how to be cells. So we have to replicate it. We have to copy it. DNA replication is called semi-conservative. Conserve means to save, and semi means sort of. So when we make a copy of the DNA, when you make the new copy, you're going to conserve part of the original copy in the new one. So when you start, you have one DNA molecule, which is double-stranded. We're going to take the 2 strands apart. And so we're going to make 2 cells. We have 2 strands. Each strand is going to get a copy of that original. And then they're going to fill in their missing strand. And they're able to do that because of those pairing rules with those bases. Okay, here's a picture to sort of help you understand it a little bit more. Okay, here's the original strand of DNA that we're going to copy. Okay, original strand is blue. So we need to copy it. So we're going to take these 2 backbones, the 2 strands, apart. One part's going to go towards this cell. The other part's going to go towards this cell because we're making 2 cells from one. Okay, so they each get an original strand from the original DNA. Okay, then they're going to fill in the bases using those pairing rules to make the second strand of DNA. This is what is meant by semi-conservative. They both get part of the original. They conserve part of the original, and then they each get a new strand that they created through the process of replication. Okay? So here's your step-by-step. First, you have to untwist the DNA because it's a twisted up ladder. So step 1 is untwist. Step 2, an enzyme is going to come in and it's going to unzip the strands. What that means is it's just going to pull apart those 2 strands by breaking those really weak hydrogen bonds that we learned about yesterday. Then, available nucleotides that are in the nucleus are going to bond to these new single strands to fill in that second strand, following the base pairing rules. Remember, if there's an A, the fill-in is going to be a T. If there's a G, then what's going to come in to fill in? A C. Okay? So remember your pairing rules. That's really important. Next, another enzyme is going to go along. Now we have 2 sets of DNA. And an enzyme is going to go and it's going to zip it back up. So it's going to hook together the bases by reforming those hydrogen bonds. And we actually need to add a number 5 here. And number 5 is retwist. So we need to twist it back up. Each and every one of these steps along the way is going to use enzymes. Remember, enzymes are proteins that help stuff happen faster. They don't change in the process. They're not a physical part of the process. They're just a helping hand along the way to help do each and every one of these steps. Make sure that you know the order of these steps and what is happening in each one. Okay, so this is the really simplified version. Okay, untwist, unzip, fill in, rezip, retwist. But of course, it's a little bit more complicated than that. Okay. What's actually going to happen is you're going to create what's called a replication bubble. Because you're not going to start at the end of the DNA and pull it apart and go to the other end. That would be too easy, right? That would be nice. But DNA is incredibly long. So that process would take too long to do it that way. So what's actually going to happen is all along the way, you're going to just pull apart the two strands randomly in the middle at several locations. And so you're going to create what's called a replication bubble, because when you pull apart the DNA in the middle, it kind of looks like there's a little bubble in there. Okay? And what's going to happen is it's going to go in both directions. Okay? And DNA can only be read in one direction, from 5 to 3. So that's called the leading strand. It can be added on very, very quickly and easily, okay, because it's reading in the correct direction, and it just pops on the new nucleotides in the correct way. Now, the other side of the DNA, though, it's not in the right direction, so we can't read it normally. What we have to do is we have to jump forwards a little bit and read back. Jump forwards a little bit and read back. Jump forwards a little bit and read back. So we're reading in the right direction. Okay? And we have to create these little sections of DNA at a time called Okazaki fragments. Okay? Those are called Okazaki fragments. Okay? And I know that this sounds a little bit confusing, so we're going to have another video or two to watch on the process so you can see it in motion. If you still don't understand at that point, call me over and ask me for help, okay? And I'll draw you a little diagram and explain it as we go, okay? Just know that you have these two strands of DNA, and they're what's called anti-parallel. They're opposites of one another. So one is basically going in this direction, and the other one is going in this direction. And as you're reading the DNA to replicate it, you can only read in one direction. Okay, that's the leading strand, read in the correct direction. The one that's going in the opposite direction, it's backwards, that's called the lagging strand, because it's a little bit slower. Okay, but let's watch a video that is going to put this in motion for us. I think that's going to make it a little bit easier to understand.